Hello, this video is going to go over Chapter 17, the endocrine system, Objective 8. Objective 8 is the factors that influence activation of the target cell by a hormone. So what factors influence the target cell? In order for a target cell to respond to a hormone, the cell must have the specific receptor for that hormone. We've kind of previously talked about that, and it's going to be on the surface of the membrane, of the plasma membrane. Or remember, if it's a lipid-based hormone, that receptor will be on the inside of the cell. A hormone receptor responds to a hormone binding by prompting the cell to perform a specific function. Hormones are molecular triggers rather than informational triggers. So they don't provide information. They, as we saw previously, result in a transformational change, conformational change, which then triggers a cascade of events within the cell. Binding of a hormone to a receptor is required. So in other words, you have to get the hormone to bind to the receptor in order for the cell, the target cell, to do anything. I mean, if the hormone's just nearby but isn't binding, then nothing's going to happen. The degree of target cell activation depends on three things. The blood levels of the hormone. Obviously, if you have more hormone available, you're going to get a quicker response. You're going to get more of a response from that target cell. There's more of a chance that hormone is going to be able to bind to the receptor if there's just more of it present. If there's less of the hormone present, then there's a less chance of that actually binding to the receptor, creating a change. Relative number of receptors for the hormone. So again, if the target cell has a lot of receptors, odds are a hormone will bind. If the target cell has fewer receptors, then there's not as many places for that hormone to bind, so the likelihood of it binding is less. And then the affinity of the binding between the hormone and the receptor. How strong is the bond between the two? If, again, that receptor is going to really grab a hold of that hormone, it's going to stick and cause changes within the cell. If the receptor doesn't bond very well, the hormone might hit it and then bounce off of it because that receptor isn't grabbing a hold of it well enough. So if that's the case, if it just kind of pops off of it because it's not able to hold it, you're not going to get any change within the cell. So it had a low affinity for that specific hormone. So with receptors, they are dynamic structures, meaning they're not stagnant. They're constantly changing. You're adding more receptors to the surface of the cell. You're decreasing the number of receptors on the cell surface. So they're constantly in flux. They're not a static kind of situation. They change based on the needs of the body. So this is how the body or the cell determines if it is needing to do something. It will either make more receptors, make it more available for a binding of a hormone, or make fewer receptors. And again, this is constantly changing. So you've got right here in this first image here, you've got a hormone right here that's kind of diamond shaped, so it will fit easily into these receptors. Target cell for hormone B, this specific cell right here will only bind to this round shaped hormone, whereas this specific cell could bind to either. So again, like we said, just because the hormone is traveling through the bloodstream doesn't mean every cell in the body is going to have a receptor for that. Now, there are a few definitions you're going to need to know. These are just straight definition kind of things. Upregulation occurs when there are persistently low levels of a hormone present, and this will cause the target cell to form additional receptors for that hormone. So if you look at this situation right here with this first cell, you can see that cell only has 2 receptors, and it's able to bind to that one little hormone that was floating around. But if you've got very little hormone present in the blood, it would be to the cell's advantage to have as many receptors as it could to bind to those few hormones that are present. If you have few hormones, few receptors, odds are it's not going to bind. If you have few hormones, you're going to have a lot of receptors, so that way it increases the chance of that cell actually binding, that receptor actually binding to that hormone, causing a change. So upregulation makes the target cell more sensitive, so it will respond to those low levels of hormones present. Downregulation is the opposite. Downregulation occurs when there's a long exposure to high levels of a hormone. And this will cause the target cell to decrease the number of receptors for that hormone. So, like in this first image right here, you've got a lot of receptors, but you've got a lot of hormone present too. That cell is constantly being triggered to do something, okay, whatever it's doing on the inside there. So, you don't need that many receptors when your hormone concentration is so high. I mean, odds are, yeah, it's going to bind if you've got that kind of situation. So in this second picture, you can see the receptors are diminishing. In this last picture, you can see there's two receptors, but there's so much hormone there that it's going to easily bind to the receptors that are present because there's just so much more hormone than there are receptors. So downregulation desensitizes the target cell, so it will respond less vigorously to the numerous amount of hormone present in the blood, preventing them from overreacting. So, hormone circulation. Hormones circulate in the blood in two forms. Now, keep in mind with the hormones, some of them are water soluble, some of them are fat soluble. So, that's going to affect those hormones and how they travel through the bloodstream. Your plasma is 92% water. Water soluble hormones are fine with that. Fat soluble hormones, not so much. So, hormones can travel freely through the bloodstream. These are going to be your water soluble hormones. Amino acid derivatives, water soluble, they can easily travel through the bloodstream. They're fine. But those ones that are lipid soluble, they're going to have to bind to a carrier protein, which will help them better transport through the watery bloodstream. So they're going to be, those hormones are going to travel bound to a protein. We'll talk more about that protein in the next chapter when we get into the components of blood. But you can see here, you've got this handout in your packet. So you've got these water soluble hormones right here. You've got this endocrine cell. It's going to secrete those water soluble hormones into the bloodstream. And you can see these yellow ones. They are very easily traveling through the bloodstream. They're going to come out, match up with the receptor on a target cell, and they're fine. Now, if you take a look at the situation below here, you've got these lipid soluble hormones. They're kind of beige shaped. Remember, these guys, they don't like water, so they're not going to like traveling through the blood where there's so much water present. So you have these blue binding proteins. I'll go ahead and give you the name. It's albumin, most likely. And that binding protein, albumin, will match up with that lipid soluble hormone, and they will travel, as you can see right here, as a pair. So you're going to travel together through the bloodstream. And then when they reach their target cell, the protein will disattach, and then that hormone will then be able to pass through the capillary into the area where that target cell is. And remember, these guys, notice the arrow right here, those lipid soluble hormones, they can easily go straight through that plasma membrane into the nucleus of the cell, which is what you see happening here. That was in the previous section. Concentration of circulating hormones. So the concentration of circulating hormones at any given time reflects the rate of release. So how much, at what speed, I guess might be a better way to say it, at what speed is that hormone being released from the endocrine gland? Is it releasing all the time? Is it releasing once a day? Is it releasing once a week? So that's what that's talking about. Number two, the speed at which it is inactivated and removed from the body. So how long is that going to travel through the bloodstream actively? It's not going to be forever. So eventually it will become inactive and no longer functioning. So the length of time for a hormone's blood levels to decrease by half is called half-life. The half-life of a hormone can vary from minutes to weeks. Like we said previously, the endocrine system's activities are going to take longer to react, but they're going to act for a longer time as opposed to the neurotransmitters in the nervous system. That's immediate. So remember when we talked previously about the definition of half-life, it is the amount of time it takes you to get from 100% of the concentration that you started at to 50% of that concentration. So I'll give you the example. similar to what we did before. Let's say you've got 100 grams of a substance in the bloodstream. How long does it take you to get to 50 grams of that? Half of what you started with. That is half-life. Water-soluble hormones have short half-lives. Remember, they're traveling through the bloodstream, not bound to a protein, not protected by a protein. They're just traveling free floating. So they're going to have shorter half-lives. Whereas lipid soluble hormones, because they're traveling through the bloodstream attached to a protein, they're going to last longer. They're going to handle going through that bloodstream a little bit better once they've attached to that protein. So this is just the handout that you have in your packet there, that's over the comparison between lipid and water soluble hormones. So I would put a star on this and definitely be able to compare and contrast the two. I've put on your Moodle page, in case you need some help with learning how to compare and contrast, how to study for that kind of question on the test. Miss Crystal has created a short video, 3 minutes long, I suppose, on some ideas on how to study for a compare and contrast type question like this. I believe she's using acid base, and what's the difference compare and contrast an acid to a base. But again, it would apply, the method of studying would apply for this content as well. So I encourage you all to go to the Moodle page underneath where I've got the tutor schedule. I've got 3 videos there that Miss Crystal has made to kind of give you some ideas on how to study for various content that you're going to run across. There is straight memorization, how you would study for just memorizing stuff, how you would study for a compare and contrast, and then how you would study for a process, kind of like what we talked about with how that lipid soluble hormone or how that water soluble hormone is going to affect that target cell. Remember, it was a series of steps, or a pathway, knowing how you get from corticotropin releasing hormone from the hypothalamus all the way to cortisol. That's a pathway. So those would be process type skills that you would need to be able to learn. She's got a video on how to study for that type of content as well to be prepared for the test. So I encourage you all to go check out those 3 videos that she's got in case you need some ideas on how to study for this type of content. 3 types of hormone interactions that are at a target cell. Again, definition type words: permissiveness, synergism, and antagonism. So hormone interaction with a target cell when it's called permissive effect. So this first bullet right here where I have that highlighted, that's the definition. So that's one that I would probably put a star beside. Make sure you know that. When the permissive effect is when the presence of one hormone enables another hormone to act. One hormone cannot act in and of itself. It has to have another hormone present in order to work. So you have right here this thyroid hormone affecting this target cell. Nothing's going to happen. You have epinephrine affecting this target cell. You have a small little bit of that target cell responding to that. But when the 2 of them act together, you're going to get that large effect that you're wanting. So one hormone may induce an increase in the number of receptors for a second hormone, resulting in an increase of the effectiveness of that second hormone. So, for instance, in our example, the thyroid hormone would cause more receptors for epinephrine to be present, which means when they act together, you're going to get a stronger effect. The synergistic effect. Again, know that definition on that first bullet. Synergistic effect, you have 2 hormones with similar effects, but together they produce an amplified effect. More than one hormone produces the same effect at the target cell, and their combined effects are amplified. So you can see right here, this green line, for example, I'm just picking one. That's epinephrine. You can see that over time, the blood glucose levels are increasing with the presence of epinephrine in the blood. You can see right here with glucagon and epinephrine, you can see the 2 combined, those 2 produce a little bit more blood glucose levels in the blood. Keep in mind, with epinephrine, you want blood glucose levels high because this is part of your fight or flight response, and those cells have got to have that glucose in order to make the necessary ATP, right, for your cells to function. So you want the glucose high during fight or flight response. But if you take a look at glucagon, epinephrine, and cortisol, Notice you see a slight rise with cortisol of blood glucose levels, but combined, all three of those, glucagon, epinephrine, and cortisol together, oh my goodness, look at that combined effect with the volume of glucose that's in the blood now. So again, this is synergistic. They're all working together to really amplify those effects. Antagonistic effect. If you know what the word means, then this definition makes a little bit of sense. Antagonistic effect is when two hormones have opposing effects. One hormone opposes the action of another hormone. In lab, you may have heard them called contrary hormones. So one hormone does one thing, the other one does the opposite. The example that we're looking at here is insulin and glucagon. So insulin secretion is going to lower blood sugar levels, where glucagon secretion is going to raise blood sugar levels. They do the opposite. Another example of two antagonistic hormones would be ones you're familiar with from AMP1, and that would be calcitonin and parathyroid hormone. Tropic versus non-tropic hormones. So make sure, again, you know the definitions of these words. Hormones can be classified based on their target cell. Again, you can classify things any which way. So these are going to be classified based on their target cell. Hormones, if they directly stimulate the target cell, there's nothing in between. In other words, the hormone gets released and goes straight to the target cell. Boom. That is called a non-tropic hormone. There's nothing else involved. However, vasopressin is an example of that. It's secreted directly from the posterior pituitary, goes straight to the kidneys, nothing in between. Okay. And then that's going to help maintain water balance in the blood. If the hormone acts on another endocrine gland, that's called a tropic hormone. So, for example, thyrotropin releasing hormone from the hypothalamus doesn't go directly to its target cell. Thyrotropin releasing hormone is going to go to the anterior pituitary, causing the release of thyrotropin, thyroid stimulating hormone, which is then going to go to the thyroid gland and stimulate the thyroid gland to release T3 and T4, your metabolic hormones. So again, that is a pathway of events. Those are tropic hormones. The example here is the hypothalamus releasing corticotropin releasing hormone, which is then going to act on the anterior pituitary to stimulate adrenocorticotropin hormone, which then goes to the adrenal glands to stimulate the release of cortisol, specifically the adrenal cortex to release cortisol. And this one right here is that visual example. So when we're talking about the pathway, I didn't really want to call it an axis. I know we're more pardon me, familiar with the word pathway than we are axis. But that's what this is, the HPA axis, which stands for H for hypothalamic, P for pituitary, A for adrenal. So it's telling you what glands are involved in this pathway. If you know the HPA axis, then you know what those letters stand for and what glands are involved. So corticotropin releasing hormone is a tropic hormone because it's acting on another gland to secrete another hormone, ACTH. So ACTH is a tropic hormone because it is acting on another gland, not the target cell, to secrete a hormone. However, cortisol, the last in this pathway, is going to go straight into the bloodstream and have wide effects on organs and tissues. So this cortisol is going to the target cell. So this is a non-tropic hormone. It's the end of the path. And that concludes objective 8.